Hi class, this is your instructor Scholar Huff. We're now in Chapter 22, The Origin of Species. Although Darwin titled his book on the origin of species, he never actually discussed what he referred to as that mystery of mysteries. How one species gives rise to another. Rather, his argument concerned evolution by natural selection. That is, how one species evolves through time to adapt to its changing environment. Although an important mechanism of evolutionary change, the process of adaptation does not explain how one species becomes another, a process we call speciation. As we shall see, adaptation may be involved in the speciation process, but it does not have to be. So before we get into how one species gives rise to another, we need to understand what a species is. Even though the definition of a species is of fundamental importance to evolutionary biology, this issue still has not been completely settled and is currently the subject of considerable research and debate. Any concept of a species must account for two phenomena: the distinctness of a species that occur together at a single locality, and the connection that exists among different populations belonging to the same species. Put out a bird feeder on your balcony, in your backyard, or wherever you may live, and you will attract a wide variety of birds. In the Midwestern United States, for example, you might routinely see cardinals, blue jays, downy woodpeckers, house finches, even hummingbirds in the summer. Although it might take a few days of careful observation, you would soon be able to readily distinguish the many different species. The reason is that species that occur together, termed sympatric, are distinctive entities that are phenotypically different, utilize different parts of the habitat, and behave differently. This observation is generally true not only for birds, but also for most other types of organisms. Occasionally, two species occur together that appear to be nearly identical. In such cases, we need to go beyond visual similarities. When other aspects of phenotype are examined, such as the mating calls or the chemicals exuded by each species, they usually reveal great differences. In other words, even though we might have trouble distinguishing them, the organisms themselves have no such difficulties. Within a single species, individuals in populations that occur in different areas may be distinct from one another. In areas where these populations occur close together, individuals often exhibit combinations of features characteristic of both populations. Look at what is called Figure 22.1, please. In other words, even though geographically distant populations may appear distinct, they are usually connected by intervening populations that are intermediate in their characteristic. For instance, you have here the eastern rat snake. Although populations at the eastern, western, and northern ends of the species' range are phenotypically quite distinct from one another, they are connected by populations labeled intergrade that are phenotypically intermediate. What can account for both the distinctness of sympatric species and the connectedness of geographically separate populations of the same species? One obvious possibility is that each species exchanges genetic material only with members of its species. If sympatric species commonly exchange genes, which they generally do not, we might expect such species to rapidly lose their distinctions as the gene pools of the different species become homogenized. Conversely, the ability of geographically distant populations of a single species to share genes through this process of gene flow may keep these populations integrated as members of the same species. Based on these ideas, in 1942, the evolutionary biologist Ernest Mayr set forth the biological species concept, which defines species as groups of actually or potentially interbreeding natural populations which are reproductively isolated from other such groups. In other words, the biological species concept says that a species is composed of populations whose members mate with each other and produce fertile offspring, or would do so if they came in contact. Conversely, populations whose members do not mate with each other or who cannot produce fertile offspring are said to be reproductively isolated. Therefore, are members of different species. What causes reproductive isolation? If organisms cannot interbreed or cannot produce fertile offspring, they clearly belong to different species. However, some populations that are considered separate species can interbreed and produce fertile offspring, but they ordinarily do not do so under natural conditions. They are still considered reproductively isolated because genes from one species generally will not enter the gene pool of the other. Table 22.1 summarizes the steps at which barriers to successful reproduction may occur. Such barriers are termed reproductive isolating mechanisms because they prevent genetic exchange between species. We will discuss examples of these next, beginning with those that prevent formation of zygotes, which are called prezygotic isolating mechanisms. Mechanisms that prevent the proper functioning of zygotes after they formed are called post-zygotic isolating mechanisms. Mechanisms that prevent formation of a zygote include ecological or environmental isolation, behavioral isolation, temporal isolation, mechanical isolation, and prevent gamete fusion. Even if two species occur in the same area, they may utilize different portions of the environment. And here is table 22.1. So even if two species occur in the same area, they may utilize different portions of the environment and thus not hybridize because they do not encounter each other. For example, lions and tigers can produce hybrid offspring in zoos. For instance, you see that before you now in figure 22.2. But even though the ranges of the two species overlapped in India until about 150 years ago, no natural hybrids were ever discovered. Lions stayed mainly in the open grassland and hunted in groups called prides. Tigers tended to be solitary creatures of the forest. Because of their ecological and behavioral differences, lions and tigers rarely came into direct contact with each other, even though their ranges overlapped over thousands of square kilometers. In another example, the ranges of two toads, Bufo woodhousei and Bufo americanus, overlap in some areas. Although these two species can produce viable hybrids, they usually do not interbreed because they utilize different portions of the habitat for breeding. Bufo woodhousei prefers to breed in streams, and Bufo americanus breeds in rainwater puddles. Similar situations occur in plants. Two species of oaks occur widely in California, the valley oak and the scrub oak. The valley oak, a graceful deciduous tree that can be as tall as 35 meters, occurs in fertile soils of open grassland, on gentle slopes, and valley floors. In contrast with the scrub oak, is an evergreen shrub, usually only 1 to 3 meters tall, which can often form a kind of dense scrub known as chaparral. The scrub oak is found on steep slopes in less fertile soils. Hybrids between these different oaks do occur and are fully fertile, but they are rare. The sharply distinct habitats of their parents limit their occurrence together, and there is little intermediate habitat where the hybrids might flourish. In Chapter 53, it describes the often elaborate courtship and mating rituals of some groups of animals. Related species of organisms, such as birds, often differ in their courtship rituals, which tends to keep these species distinct in nature, even if they inhabit the same places. Sympatric species avoid mating with members of the wrong species in a variety of ways. Every mode of communication imaginable appears to be used by some species. Differences in visual signals, as just discussed, are common. However, other types of animals rely more on other sensory modes for communication. Many species, such as frogs, birds, and a variety of insects, use sound to attract mates. Predictably, sympatric species of these animals produce different calls. Similarly, the songs of lacewings are produced when they vibrate their abdomens against the surface on which they are sitting, and sympatric species produce different vibration patterns. So behavioral isolation is real. Other species rely on detection of chemical signals called pheromones. The use of pheromones in moths has been particularly well studied. When female moths are ready to mate, they emit a pheromone that males can detect at great distances. Sympatric species differ in the pheromone they produce. Either they use different chemical compounds, or if they are using the same compounds, the proportions used are different. Laboratory studies indicate that males are remarkably adept at distinguishing the pheromones of their own species from those of other species, or even from synthetic compounds similar, but not identical to that of their own species. Some species even use electroreception. African and South Asian electric fish independently have evolved specialized organs in their tails that produce electrical discharges, and electroreceptors on their skins detect them. These discharges are used to communicate in social interactions. Field experiments indicate that males can distinguish between signals by their own and other co-occurring species, probably on the basis of differences in the timing of the electrical impulses. There you have it, behavioral isolation, be it visual signals, production of sound, pheromones, or electroreception. Temporal isolation. Two species of wild lettuce grow together along roadsides throughout the southeastern United States, Latuca graminifolia and Latuca canadensis. Hybrids between these two species are easily made experimentally and are completely fertile, but these hybrids are rare in nature because Latuca graminifolia flowers in early spring and Latuca canadensis flowers in summer. When their blooming periods overlap, as happens occasionally, the two species do form hybrids which may become locally abundant. Many species of closely related amphibians have different breeding seasons that prevent hybridization. For example, 5 species of frogs in the genus Rana occur together in most of the eastern United States, but hybrids are rare because the peak breeding time is different for each of them. Mechanical isolation. Structural differences prevent mating between some related species of animals. Aside from such obvious features as size, the structures of the male and female copulatory organs may be incompatible. In many insect and other arthropod groups, the sexual organs, particularly those of the male, are so diverse that they are used as a primary basis for distinguishing species. Similarly, flowers of related species of plants often differ significantly in their proportions and structures. Some of these differences limit their transfer of pollen from one plant species to another. For example, bees may carry the pollen of one species on a certain place on their bodies. If this area does not come in contact with the receptive structures of the flowers of another plant species, the pollen is not transferred. Prevention of gamete fusion. In animals that shed gametes directly into water, the eggs and sperm derived from different species may not attract or fuse with one another. Many animal species that have internal fertilization may not hybridize successfully because sperm of one species functions so poorly with the reproductive tract of another that fertilization never takes place. In plants, the growth of pollen tubes may be impeded in hybrids between different species. In both plants and animals, isolating mechanisms such as these prevent the union of gametes, even following successful mating. All the factors we have discussed so far tend to prevent hybridization. If hybrid mating does occur and zygotes are produced, many factors still prevent those zygotes from developing into normal, functioning, fertile individuals. As you saw in Chapter 19, development is a complex process. In hybrids, the genetic complements of two species may be so different that they cannot function together normally in embryonic development. For example, hybridization between sheep and goats produces embryos that die in the earliest developmental stages. The leopard frogs of the eastern United States are a group of similar species assumed for a long time to constitute a single species. Please see figure 22.5, shown to you all here. Careful examination, however, revealed that although the frogs appear similar, successful mating between them is rare because of problems that occur as the fertilized eggs develop. Many of the hybrid combinations cannot be produced even in laboratory. Examples of this kind in which the existence of multiple species has been recognized only as a result of hybridization experiments are common in plants. Even when hybrids survive the embryo stage, they still may not develop normally. If the hybrids are less physically fit than their parents, they will almost certainly be eliminated in nature. Even if a hybrid is vigorous and strong, as in the case of the mule, which is a hybrid between a female horse and a male donkey, it still may be sterile and thus incapable of contributing to succeeding generations. Hybrids may be sterile because of the development of sex organs is abnormal, because the chromosome number derived from the respective parents cannot pair properly during meiosis or due to a variety of other causes. In the case of the mule, for example, the parental species have different chromosome numbers. I repeat, they have a different number of chromosomes, and as a result, the mule's chromosomes cannot align properly during meiosis, thus rendering the animal infertile. The biological species concept has proved to be an effective way of understanding the existence of species in nature. Nonetheless, it fails to take into account all observations, leading some biologists to propose alternative species concepts. One criticism of the biological species concept concerns the extent to which all species truly are reproductively isolated. By definition, under the biological species concept, species should not be able to interbreed and produce fertile offspring. But in recent years, biologists have detected much greater amounts of interspecific, i.e., between species hybridization that was previously thought to occur between populations that seem to coexist as distinct biological entities. Botanists have always been aware that plant species often undergo substantial amounts of hybridization. More than 50% of California plant species included in one study, for example, were not well defined by genetic isolation. This coexistence without genetic isolation can be long-standing. Fossil data show that balsam poplars and cottonwoods have been phenotypically distinct for 12 million years, but they also have been routinely produced hybrids throughout this time. Consequently, many botanists have long felt that biological species concept is misnamed and applies only to animals. New evidence, however, increasingly indicates that hybridization is not at all that uncommon in animals either. In recent years, many cases of substantial hybridization between animal species have been documented. One survey indicated that almost 10% of the world's more than 10,000 bird species are known to have hybridized in nature. The Galapagos finches provide a particularly well-studied example. 3 species on the island of Daphne Major, the medium ground finch, the cactus finch, and the small ground finch, are clearly distinct morphologically, and they occupy different ecological niches. Studies over the past 30 years by Peter and Rosemary Grant found that on average 2% of the medium ground finches and 1% of the cactus ground finches mated with other species every year. Furthermore, hybrid offspring appeared to be at no disadvantage in terms of survival or subsequent reproduction. This is not a trivial amount of genetic exchange. And one might expect to see the species coalesce into one genetically variable population, but the species are maintaining their distinctness. Hybridization is not rampant throughout the animal world, however. Most bird species do not hybridize, and probably even fewer experience significant amount of hybridization. Still, hybridization is common enough to cast doubt on whether reproductive isolation is the only force maintaining the integrity of species. An alternative species concept, the ecological species concept, proposes that distinctions among species are maintained by natural selection. The idea that each species has adapted to its own specific part of the environment, stabilizing selection, described in Chapter 20, then maintains the species' different adaptations. Hybridization has little effect because alleles introduced into one species' gene pool from the other species are quickly eliminated by natural selection. And you probably recall from Chapter 20 that the interaction between gene flow and natural selection can have many outcomes. In some cases, strong selection can overwhelm any effects of gene flow. This is how natural selection can maintain species' differences in the face of gene flow, but in other situations, gene flow can prevent natural selection from eliminating less successful alleles from a population. In this case, the biological species concept would be applicable. The biological species concept has been criticized for other reasons as well. For example, it can be difficult to apply the concept to populations that are geographically separated in nature because the individuals of these populations do not encounter each other. It is not possible to observe whether they would interbreed naturally. Although experiments can determine whether fertile hybrids can be produced, this information is not enough. Many species that coexist without interbreeding in nature will readily hybridize in artificial settings of the laboratory or zoo. Consequently, evaluating whether such populations constitute different species is ultimately a judgment call. In addition, the concept is more limited than its name would imply. Many organisms are asexual and reproduce without mating. Reproductive isolation, therefore, has no meaning for such organisms. For these reasons, a variety of other ideas have been put forth to establish criteria for defining species. Many of these are specific to a particular type of organism, and none has universal applicability. In reality, there may be no single explanation for what maintains the identity of species. Given the incredible variation evident in plants, animals, and microorganisms in all aspects of their biology, it would not be surprising to find that different processes are operating in different organisms. In addition, some scientists have turned from emphasizing the process that maintains species distinctions to examining the evolutionary history of populations. These genealogical species concepts are currently the topic of great debate and discussed in the next chapter. One of the oldest questions in the field of evolution is, how does one ancestral species become divided into two descendant species? If species are defined by the existence of reproductive isolation, then the process of speciation is identical to the evolution of reproductive isolating mechanisms. The formation of species is a continuous process, and as a result, two populations may have gone only part of the way toward becoming different species and thus only be partially reproductively isolated. For example, because of the behavior or the ecological differences, organisms of two populations may be more likely to mate with members of their own population, but between population matings may still occur. If mating occurs and fertilization produces a zygote, post-zygotic barriers may also be incomplete. Developmental problems may result in lower embryo survival or reduced fertility, but some individuals may survive and reproduce. What happens when two populations come in contact thus depends on the extent to which isolating mechanisms have already evolved. If isolating mechanisms have not evolved at all, then the two populations will interbreed freely, and whatever differences have evolved between them should disappear over the course of time as genetic exchange homogenizes the populations. Conversely, if the populations are completely reproductively isolated, then no genetic exchange will occur, and the two populations will remain different species. More interesting is the situation in which hybrids are produced that are either only partly sterile or not as well adapted to the existing habitats as their parents. Selection would favor many alleles on the parental populations that prevented hybridization, because individuals that did not engage in hybridization would produce more successful offspring. The result would be that selection would improve prezygotic isolating mechanisms until two populations were completely reproductively isolated. This process is termed reinforcement, because initially incomplete isolating mechanisms are reinforced by natural selection until they are completely effective. An example of reinforcement is provided by wildflowers, the wildflower called phlox. You'll see that in figure 22.6 and shown for you all now. Most phlox flowers are pale blue color, sometimes with a pink tinge. Drummond's phlox is a beautiful species that grows profusely along highways. It is no exception. The one exception is in certain parts of Texas, where it is sympatric with other species, the pointed phlox. When the two species are found together, the pointed phlox maintains a pale blue coloration, but Drummond's phlox is a deep shade of red. How has this color difference evolved, and why? The color of these flowers is a result of the red and blue color pigments produced by different genes and leading to the light blue, slightly pinkish color, and another gene that affects the amount of all pigments produced, affecting the intensity of the color. In sympatric populations of Drummond phlox, an allele that causes an increase in pigment production has been replaced by the normal allele. By itself, this change would lead to an increase in production of both red and blue pigments, leading to a deeper shade of the same color, a much deeper shade. However, a new allele at a second gene has knocked out production of the blue pigment, leaving the red pigment to predominate. This results in the deep red color of the sympatric Drummond's phlox flowers. But why has this change occurred? The answer lies in the interaction between the two phlox species. The flowers are able to interbreed, but their offspring have very low fertility. Consequently, any factor that would diminish interbreeding would be favored by natural selection. And that's just what color does. Both species are pollinated by butterflies. They go from one flower to another, and it turns out that individual butterflies have color preferences. As a result, some butterflies visit only the light blue flowers, and others visit only the dark red ones, and thus pollen from one species do not fertilize flowers from the other species. This example illustrates how reproductive isolation, initially resulting from post-zygotic infertility, can be reinforced by the evolution of pre-mating isolation. Reinforcement is not inevitable, however. When incompletely isolating populations come together, gene flow immediately begins to occur between them. Although hybrids may be inferior, they are not completely inviable or infertile. If they are, the species would already be completely reproductively isolated. When the surviving hybrids reproduce with the members of either population, they will serve as a conduit of genetic exchange from one population to the other, and the two populations will tend to lose their genetic distinctiveness. Thus, a race ensues. Can complete reproductive isolation evolve before gene flow erases the differences between the populations? The outcome depends on the initial conditions and the particular history of the species involved, but many experts consider reinforcement to be the less common outcome. What role does natural selection play in the speciation process? Certainly the process of reinforcement is driven by natural selection, favoring the evolution of complete reproductive isolation, but reinforcement may not be common. In situations other than reinforcement, does natural selection play a role in the evolution of reproductive isolating mechanisms? As mentioned in Chapter 20, populations may diverge as a result of genetic drift, random change in small populations, the founder effect, and population bottlenecks all may lead to changes in traits that cause reproductive isolation. For example, in the Hawaiian Islands, closely related species of Drosophila often differ greatly in their courtship behavior. Colonization of new islands by these fruit flies probably involved a founder effect in which one or a few flies, perhaps only a single pregnant female, were blown by strong winds to the new island. Changes in courtship behavior between ancestor and descendant populations may be the result of such founder events. Given enough time, any two isolated populations will diverge because of genetic drift. In some cases, this random divergence may affect traits responsible for reproductive isolation, and speciation may occur. Although random processes may sometimes be responsible, in many cases, natural selection probably plays a role in the speciation process. As populations of a species adapt to different circumstances, they likely accumulate differences that may lead to reproductive isolation. It is important to realize that in cases like this, contrary to what occurs during reinforcement, natural selection isn't acting in favor of those individuals that avoid hybridizing with other species. Rather, increased reproductive isolation evolves as an incidental consequence of the evolution of different adaptations in different populations. For example, if the population of flies adapts to wet habitats and another to dry areas, then natural selection will favor a variety of corresponding differences in physiological and sensory traits. These differences may produce ecological and behavioral isolation and may cause any hybrids the two populations produce to be poorly adapted to either habitat. Selection on mating behavior might also incidentally lead to the evolution of reproductive isolation. Male anolis lizards, for example, court females by extending a colorful flap of skin called the dewlap located under their throats. See Figure 22.7. The ability of one lizard to see the dewlap of another lizard depends not only on the color of the dewlap, but on the environment in which the lizards live. A light-colored dewlap is most effective in reflecting light in a dim forest, whereas dark colors are more effective or more apparent in the bright glare of open habitats. As a result, when these lizards occupy new habitats, natural selection favors the evolutionary change in the dewlap color because males whose dewlaps cannot be seen will not attract any mates. But the lizards also distinguish members of their own species from other species by the color of the dewlap. Adaptive change in mating signals in new environments to enhance visibility to conspecifics could therefore produce reproductive isolation from populations in the ancestral environment if individuals from the two populations ever come into contact. Because of differences in their dewlap colors, they might not recognize each other as members of the same species. Laboratory scientists have conducted experiments on fruit flies and their fast-reproducing organisms and other fast-reproducing organisms, in which they isolate populations in different laboratory chambers and measure how much reproductive isolation involves. These experiments indicate that genetic drift by itself can lead to some degree of reproductive isolation, but in general, reproductive isolation evolves more rapidly when populations are forced to adapt to different laboratory environments. So although natural selection in the experiment does not directly favor traits because they led to reproductive isolation, the incidental effect of adaptive divergence is that populations in different environments become reproductively isolated. For this reason, some biologists believe the term isolating mechanisms is misguided because it implies that the traits evolved specifically for the purpose of genetically isolating a species, which in most cases, except reinforcement, is probably incorrect. Speciation is a two-part process. First, initially identical populations must diverge, and second, reproductive isolation must evolve to maintain these differences. The difficulty with this process, as we have seen, is that the homogenizing effect of gene flow between populations is constantly acting to erase differences that may arise by genetic drift or natural selection. Gene flow occurs only between populations that are in contact, however, and populations can become geographically isolated for a variety of reasons. Consequently, evolutionary biologists have long recognized that speciation is much more likely in geographically isolated populations. And you can see that here in figure 22.8. So it might be that there is that geographic barrier there. You see that there's a stream there, or maybe a small river, or even there's a valley there between the mountain range, between those two mountains. Nonetheless, let's begin. Ernest Mayr was the first biologist to demonstrate that geographically separated or allopatric species, or allopatric populations, appear much more likely to have evolved substantial differences leading to speciation. Marshalling data from a wide variety of organisms and localities, Mayr made a strong case for allopatric speciation as a primary means of speciation. For example, little paradise kingfisher varies little throughout its wide range in New Guinea, despite the great variation on the island's topography and climate. By contrast, isolated populations on nearby islands are strikingly different from one another and from mainland population, which is shown to you here. Thus, geographic isolation seems to have been an important prerequisite for the evolution of differences between populations. Many other examples indicate that speciation can occur under allopatric conditions, because we would expect isolated populations to diverge over time by either drift or selection. This result is not surprising. Rather, the more intriguing question becomes, is geographic isolation required for speciation to occur? For decades, biologists have debated on whether one species can split into two at a single locality, without the two new species ever having been geographically separated. Investigators have suggested that this sympatric speciation could occur either instantaneously or over the course of multiple generations. Although most of the hypotheses suggested so far are highly controversial, one type of instantaneous sympatric speciation is known to occur commonly as a result of polyploidy. Instantaneous sympatric speciation occurs when an individual is born that is reproductively isolated from all other members of its species. In most cases, a mutation would cause an individual to be greatly different from the others of its species, would have many adverse side effects due to the many pleiotropic effects of most genes, and the individual likely would not survive. One exception, often seen in plants, however, occurs to the process of polyploidy, which produces individuals that have more than two sets of chromosomes. Polyploid individuals can arise in two ways. Autopolyploidy, all of the chromosomes come from a single species. This might happen, for example, due to an error in cell division that causes a doubling of chromosomes. Such individuals are termed tetraploids because they have 4 sets of chromosomes, can self-fertilize or mate with other tetraploids, or can mate with other tetraploids, but cannot mate or produce fertile offspring with normal diploids. The reason that the tetraploid species produce The reason is that tetraploid species produce diploid gametes that produce triploid offspring, which combine with haploid gametes from normal diploids. Triploids are sterile because the odd number of chromosomes prevents proper pairing during meiosis. A more common type of polyploid speciation is allopolyploidy, which may happen when two species hybridize. The resulting offspring have one copy of the chromosomes from each species, is usually infertile because chromosomes do not pair correctly in meiosis. However, such individuals are often otherwise healthy. Sometimes the chromosomes of such an individual spontaneously double, just as described for autopolyploidy. Consequently, the resulting tetraploid has two copies of each set of chromosomes, and pairing during meiosis is no longer a problem. As a result, tetraploids are able to interbreed with other similar tetraploids, and a new species has been created. It is estimated that about half of the approximate 260,000 species of plants have a polyploid episode in their history, including many of great commercial importance, such as bread, wheat, cotton, tobacco, sugarcane, bananas, and potatoes. Speciation by polyploidy is also known to occur in a variety of animals, including insects, fish, and salamander, although much more rarely than in plants. Sympatric speciation. So some investigators believe that sympatric speciation can occur over the course of multiple generations through the process of disruptive selection. As noted back in Chapter 20, disruptive selection can cause a population to contain individuals exhibiting two different phenotypes. One of the most visible manifestations of evolution is the existence of groups of closely related species that have recently evolved from a common ancestor and have adapted to many different parts of the environment. These adaptive radiations are particularly common in situations in which a species occurs in an environment with few other species and many available resources. One example is the creation of new islands through volcanic activity, such as the Hawaiian and Galapagos Islands. Another example is a catastrophic event leading to the extinction of most other species, a phenomenon known as mass extinction, which will be discussed later in this chapter. Adaptive radiation can also result when a new trait called a key innovation evolves within a species, allowing it to use resources or other aspects of the environment that were previously inaccessible. Classic examples of key innovation leading to adaptive radiation are the evolution of lungs in fish and wings in birds and insects, both of which allow descendant species to diversify and adapt to many newly available parts of the environment. Adaptive radiation requires both speciation and adaptation to different habitats. A classic model postulates that a species colonizes multiple islands in an archipelago. Speciation subsequently occurs allopatrically, and then the newly arisen species colonize other islands, producing multiple species per island, as you see coming up in a moment. In Figure 22.11, the ancestral species flies from the mainland to colonize. The ancestral species spreads to different islands. Populations on different islands evolve to become different, distinct species. The species evolves differently to adaptations in allopatry. Colonization of islands continues, and species evolved different adaptations to minimize competition with other species. Adaptations to new habitats can occur either during the allopatric phase, as the species respond to different environments on the islands, or after two species become sympatric. In the latter case, this adaptation may be driven by selective pressures to minimize competition for available resources with other species. In this process, termed character displacement, two reproductively isolated but ecologically similar species come in contact. Because the two species use the same resources, natural selection in each species favors those individuals that use resources not used by the other species. Because those individuals will have greater fitness, whatever traits cause differences in resource use will increase in frequency, and over time, the species will diverge. Figure 2212 shows this. One example of character displacement involves the three-spined sticklebacks, a small fish found in temperate lakes throughout the northern hemisphere. In British Columbia, many of those lakes were created in the past 1,200 years, when the glaciers melted at the end of the Ice Age. Some of these lakes near the ocean have been invaded by marine populations of stickleback. These fish tend to occur in habitats throughout the lake and evolved to be different from their marine ancestors. So different, in fact, that the few lakes in which a second colonization occurred, the two stickleback populations had evolved a high degree of reproductive isolation. As a result, the natural selection led to the evolution of differences in the two populations to minimize competition for food. One stickleback adapted to using open water by evolving a streamlined body form for greater speed and larger gill structures for more effectively straining out the zooplankton that occur in the open. At the same time, the other population adapted to foraging near the margins and the bottom of the lake, evolving a larger starter body size and altering the shape of the mouth to better feed on larger invertebrates from the lake floor. Similar character displacement occurred in 7 doubly colonized lakes. And you can see those sticklebacks in figure 2313. Here is figure 2313 showing those two different species of stickleback. More than 1,000 species of fly in the genus Drosophila occur on the Hawaiian Islands. New species of Drosophila are still being discovered in Hawaii, although the rapid destruction of the native vegetation is making the search more difficult. Aside from their sheer number, Hawaiian Drosophila species are unusual because of their incredible diversity of morphological and behavioral traits. If you can look there at figure 2214. When their ancestors first reached the islands, they encountered many empty habitats that other kinds of insects and other animals occupied elsewhere. As a result, the species have adapted to all manners of fruit fly life and include predators, parasites, and herbivores, as well as species specialized for eating detritus in leaf litter and the nectar of flowers. The larvae of various species living in rotting stems, fruits, bark, leaves, or roots, or feed on sap. No comparable diversity of Drosophila species is found anywhere else in the world. The great diversity of Hawaiian island species is a result of the geographical history of these islands. New islands have continually arisen from the sea in this region. As they have done so, they have appeared to be invaded successively by the various Drosophila groups present on the older islands. New species thus have evolved as new islands have been colonized. In addition, the Hawaiian Islands are among the most volcanically active islands in the world. Periodic lava flows have created patches of habitat within an island surrounded by a sea of barren rock. These land islands are termed kippukas, and then Drosophila populations isolated in these kippukas often undergo speciation. In these ways, rampant speciation combined with ecological opportunity has led to unparalleled diversity of insect life. So ancestor of Darwin's finches reaches the Galapagos Islands before the other land birds. So they were subjected to different selective pressures as they adopted new lifestyles and geographic isolation on many islands. So there is now a diverse population, and some evolved into separate species, and they're occupying many different habitats. So the differences between species likely resulted from character displacement, as initially similar species diverged to minimize competitive pressures. So you have the ground finches and the cactus finches, the tree finches, the tree vegetarian finches, and even the warbler finches. And these show differing types of finches from the Galapagos Islands. Let's now speak briefly to cichlid fishes in Lake Victoria. So Lake Victoria is an immense, shallow freshwater sea about the size of Switzerland in the heart of the equatorial East Africa. Until recently, the lake was home to an incredibly diverse collection of over 450 species of cichlid fishes. So the cichlids, they are small perch-like fishes ranging from 5 to 25 centimeters in length, and the males come in endless varieties of colors. The ecological and morphological diversity of these fish is remarkable, particularly given the short span of time in which they've evolved. So we can gain some sense of the vast range of types by looking at how different species eat. So there are the mud biters, the algae scrapers, the leaf chewers, the snail crushers, the zooplankton eaters, the insect eaters, the prawn eaters, and the fish eaters. So of all of this, the cichlid fish have a remarkable key innovation that have been instrumental in their evolutionary radiation. They carry a second set of functioning jaw. So this trait in many other fish, but cichlids, it is greatly enlarged. So their ability of this second jaw to manipulate and process food has freed the oral jaws to evolve for other purposes, and the result has been the incredible diversity of the ecological roles filled by these fish. So over the last several decades, from the 1950s, the Nile perch were introduced, and as such, 70% of those fishes, of cichlids, have gone extinct. And this shows the second set of jaws in the back, and this is figure 22.17a, and you're seeing just a bit of diversity of those cichlids there. We've discussed the manner in which speciation may occur, but we haven't yet considered the relationship between speciation and the evolutionary change that occurs within a species. Two hypotheses: gradualism and punctuated equilibrium. For more than a century after the publication of On Origin of Species, the standard view was that evolution occurred very slowly. Such change would be nearly imperceptible from generation to generation, but would accumulate such that over the course of thousands and millions of years, major changes could occur. This view is termed gradualism. An alternative possibility is that species experience long periods of little or no evolutionary change, which is known as stasis, punctuated by short bursts of evolutionary change over geologically short time intervals. This is termed punctuated equilibrium. Some have argued that these periods of rapid change occurred only during speciation process. We have seen in chapters 20 and 21 that when natural selection is strong, the evolutionary change can occur rapidly. So the punctuated part of the theory is not controversial. A more difficult question involves the long periods of stasis, the equilibrium. Why would species exist for thousands or even millions of years without changing? Although a number of possible reasons have been suggested, most researchers now believe that a combination of stabilizing and oscillating selection is responsible for stasis if the environment does not change over long periods of time, or if environmental changes oscillate back and forth, then stasis may occur for long periods. One factor that may enhance this stasis is the ability of species to shift their ranges. For example, during the ice ages, when the global climate cooled, the geographic ranges of many species shifted southward so that the species continued to experience similar environmental conditions. The fossil record shows that some well-documented groups such as African mammals clearly have evolved gradually, not in spurts. Other groups such as marine bryozoa seem to show an irregular pattern of evolutionary change predicted by the punctuated equilibrium model. It appears, in fact, that gradualism and punctuated equilibrium are two ends of a continuum. Although some groups have evolved solely in a gradual manner, others only in a punctuated mode. Many other groups show evidence of both gradual and punctuated episodes at different times in their evolutionary history. The idea that speciation is necessarily linked to phenotypic changes has not been supported. However, so on one hand, it is now clear that speciation can occur without substantial phenotypic change. For example, closely related salamander species are nearly indistinguishable. On the other hand, it is clear that phenotypic change can occur within species in the absence of speciation. So here at figure 22.18a is showing you all gradualism slowly through time and is not linked to speciation, whereas figure 22.18b is showing you all punctuated equilibrium, requiring that phenotypic change occurs in bursts associated with speciation, separated by long periods of little change or even no change. Biological diversity has increased vastly over the last 600 million years, but the trend has been far from consistent. After a rapid rise, diversity reached a plateau for about 200 million years, but since then has risen steadily, because changes in the number of species reflected the rate of origin of new species relative to the rate at which existing species disappear. This long-term trend reveals that speciation has, in general, surpassed extinction. Nonetheless, speciation has not always outpaced extinction, and particularly interspersed in the long-term increase in species diversity may have been sharp declines termed mass extinctions. 5 mass extinctions have been identified, the most severe occurring at the end of the Permian period, approximately 250 million years ago. At that time, more than half of all plants and animal families, as many as 96% of all species, may have perished, as you can see. The most famous and well-studied extinction, although not as drastic, occurred at the end of the Cretaceous period, 66 million years ago, which at the time, the dinosaurs, except for birds, which are one type of dinosaur, and a variety of other organisms, went extinct. Recent findings have supported the hypothesis that this extinction event was triggered when a large asteroid slammed into the Earth, perhaps causing global forest fires, obscuring the sun for months by throwing particles into the air. The cause of other mass extinction events is less certain. Some scientists suggest that asteroids may have played a role in at least some of the other mass extinction events. Other hypotheses implicate global climate change, mass volcanic eruptions, and other causes. One important result of mass extinctions is that not all groups of organisms are affected equally. For example, in the extinction at the end of the Cretaceous, not only dinosaurs, but also marine and flying reptiles and ammonites, a type of mollusk, went extinct. Marsupials, flowering plants, birds, and some forms of plankton were greatly reduced in diversity. In contrast, turtles, crocodilians, and amphibians seem to have been unscathed. Why some groups were harder hit than others is not clear. But one hypothesis suggests that survivors were those animals who could shelter underground or in water, and that either could scavenge or required little food in the cool temperatures that resulted from the blockage of sunlight. A consequence of mass extinctions that previously dominant groups may perish, thus changing the course of evolution. This is certainly true of the Cretaceous extinction. During the Cretaceous period, placental mammals were a minor group composed of species that were mostly no larger than a house cat. The dinosaurs, which had dominated the world for more than 100 million years, disappeared at the end of this period. The placental mammals underwent a significant adaptive radiation. It is humbling to think that humans might never have arisen had that asteroid not struck Earth 65 million years ago. I hope you all heard that. As the world around us illustrates today, species diversity does rebound after mass extinctions, but this recovery is not rapid. Examination of the fossil record indicates that the rates of speciation do not immediately increase after an extinction pulse, but rather about 10 million years to reach the maximum. The cause of this delay is not clear, but it may result because it takes time for ecosystems to recover and for the process of speciation and adaptive diversification to begin. Consequently, species diversity may require 10 million years or even much longer to attain its previous level. Right now, a sixth mass extinction is underway. The number of species in the world in recent times is greater than it has ever been. Unfortunately, that number is decreasing at an alarming rate due to human activity. See Chapter 58. Some estimate that as much as 1/4 of all species will become extinct in the near future, a rate of extinction not seen on Earth since the Cretaceous mass extinction. Moreover, the rebound in species diversity may be even slower than following previous mass extinction events, because instead of the ecologically impoverished but energy-rich environment that existed after previous mass extinctions, a large part of the world's resources will be taken up by human activities, leaving few resources available for adaptive radiation. This has been your instructor, Scholar Huff. Please let me know if you have any questions. Thank you all for learning.